An important systemic component of inflammation and innate immunity in general is fever. Fever is noted as an abnormally elevated body temperature. Sometimes we'll have fevers from a known origin, sometimes from an unknown origin. They're called FUOs when they're from unknown origin. Fever is a nearly universal symptom of infections, but it's also associated with certain allergies, as well as some cancers. Body temperature is normally regulated by the hypothalamic control center in the brain. This is essentially a thermostat that regulates heat production, and it only varies very slightly by a degree or so throughout the course of a day. When the body is exposed to a pyrogen, which is anything that causes fever, it sets the hypothalamic thermostat to a higher setting by signaling the muscles to increase their heat production and signaling peripheral arterioles to decrease heat loss through vasoconstriction. These pyrogens can either be exogenous, like blood products or vaccines, or endogenous, things that are liberated by monocytes or neutrophils or macrophages in the process of phagocytosis. Now, fever has some benefit, other than it being a sign of infection. It inhibits the multiplication of temperature-sensitive microorganisms like the poliovirus or the cold virus, many other viruses. When you consider this, why would you want to take an antipyretic drug like Tylenol or aspirin to reduce the fever? Because if we reduce fever, surely are we not encouraging the growth of these pathogens? Fever also impedes the nutrition of bacteria by reducing the availability of iron. During a fever, macrophages will stop releasing iron from some of their iron-greedy proteins where they're storing it. So pathogens have a very limited source of iron, and if they can't get it from us, they can't get it. Also, fever increases metabolism and stimulates immune reactions and naturally protective physiological processes. For example, it speeds up hematopoiesis, phagocytosis, as well as specific immune reactions. So should we really be suppressing fever? Now, moderate fever in healthy individuals should probably run its course. However, if you have a high or very prolonged fever, this can be potentially dangerous, especially if the individual has some other underlying circumstances. So it should be treated immediately. So in our second line of defense, we had inflammation, phagocytosis, interferons, and then the complement system. So now let's take a look at antimicrobial proteins involving interferon. Interferon is produced naturally by certain white blood cells like lymphocytes and fibroblasts and macrophages, as well as some tissue cells. It's involved in wars against viruses, some other microbes, and certainly in immune regulation and intercommunication between cells is the key. There are three major types of interferon. There's alpha interferon, there's beta interferon, and gamma interferon. All three classes of these interferons are produced in response to viruses, RNA, as well as immune products and various other antigens. They bind to cell surfaces and induce changes in genetic expression. We'll watch an animation here shortly. Interferon alpha and beta also stimulate phagocytes, and interferon gamma is an immune regulator of macrophages and the T and B cells. One thing that I find most interesting is all of the interferons can inhibit the expression of cancer genes and have tumor suppressor effects. So interferon is often involved in cancer treatments. Here's an example of how interferon works. If a virus is taken into a cell and infects it, it stimulates production of the interferon gene. If interferon is produced through transcription and translation, this interferon then will travel out of the cell. Now the interferons are in the extracellular environment, and they can stimulate other cells. So the other cells in the environment have special receptors for the interferons. Once they get that signal, it'll signal activation of genes in their nucleus, and they'll synthesize antiviral proteins. These antiviral proteins don't prevent viruses from entering the cell, but it does make the cell very prepared to fight viruses when they do enter the cell. so they can degrade the viruses and their nucleic acids and hopefully prevent further viral infections. So interferon you may also have heard in use of therapy for AIDS patients. Let's take a quick look at an animation to see this process in action.
When a cell is infected by a virus, the virus enters the cell and produces structures that are not found in uninfected cells. The presence of this viral material signals the cell to produce interferon. The interferon moves out of the cell and attaches to receptors on nearby cells of the same type. The cell that produces the interferon is unable to save itself. The virus replicates in this cell and then moves out to infect nearby cells. The nearby cell that already has interferon bound to its surface responds in several ways, including production of enzymes that degrade messenger RNA and prevent protein synthesis. Thus, a virus can attach and enter the cell, but completion of the viral replication cycle is prevented.
Let's have a quick concept check. A patient's serum is found to have elevated interferon levels. What is the most probable cause? A fungal infection, a bacterial infection, a viral infection, or no infection at all? The patient's normal. Well, we just learned that interferon levels increase primarily with viral infections, although we may see some increase with bacterial and fungal infections. Definitely, they're a sign of viral infections. Now we move on to looking at the complement system. The complement system is an incredibly complex system. We're just going to try and brush the surface here without confusing you. The complement system consists of at least 26 blood proteins that work in concert to destroy bacteria and certain viruses. It was named the complement system because it complements the immune system. It's a cascade reaction. A cascade reaction means that one thing causes another thing, until we eventually get an end result that we're interested in. There are 3 different pathways that yield all similar end results: the classical pathway, the lectin pathway, and the alternative pathway. You won't be required to understand the details of each of these pathways, just that we have a whole system of proteins that are in the blood that really help with invading microbes. The classical pathway is activated by the presence of antibodies bound to microorganisms. The lectin pathway is activated when the host serum proteins bind to a sugar that's found on fungal cell walls. And the alternative pathway is activated when complement proteins bind to the normal cell wall and surface components of microbes. And they do some really interesting things. Let's look at this cascade for a moment. The whole initiation is because the microbes or an antibody react with the first chemical component in the pathway. As you can see from this figure here, the initial chemical component could be a number of different things, and each has a number of different complement proteins involved in the process. But eventually we end up with the same result. We'll have amplification. Simply put, this complex activates a number of different proteins. So we're amplifying in the cascade. One thing causes the activation of 3 others, each of those cause the activation of 3 others, so on and so forth. In polymerization, we have reactions that have a successful assembly of an attack complex. And then there's the membrane attack. Literally, we have a set of proteins that come together to digest holes in the membrane of bacterial cells, as well as in enveloped viruses. So the whole point of this complement system is that a bunch of different proteins come together through 3 different pathways that we've seen here, and they cause a formation of a ring structure of proteins that punches holes in the cell membranes. Once we've punched holes in the cell membranes, the contents leak out and that cell is lysed. So the complement cascade is a way for proteins to come together in order to blow holes in the membranes of both invading bacterial cells as well as enveloped viruses. Let's take a look at some of the specifics of the cascade. However, you do not need to know these specifics, just so you can see how it all works.
Regardless of whether the complement system is activated by the classical, alternative, or lectin pathway, The activated complement proteins are involved in several important processes associated with host defenses, including inflammation, attraction of neutrophils, opsonization, and cell lysis. Complement C3a participates in inflammation by inducing changes that cause the walls of local blood vessels to become more permeable. This facilitates movement of phagocytic cells from the circulation into the tissues. Complement 5a induces a directed chemotactic migration of phagocytes to the site of complement activation. Complement C3b functions as an opsonin. It binds to microbial cells, making them more susceptible to phagocytosis. Complement proteins C5b, C6, C7, C8, and C9 react together to form a hole in the cell membrane. When C5 convertase converts C5 into C5a and C5b, the C5b binds to the cell surface and combines with C6. C5b6 binds C7, enabling it to insert into the membrane. Binding of C8 is followed by binding of several C9s. The hole generated by formation of this membrane attack complex causes the cell to lyse.
Finally, a third set of antimicrobial proteins, the iron-binding proteins. Iron is important for both humans and bacteria in order for us to accomplish all of our metabolic needs. Now, we have several molecules that bind to iron, specifically to lessen the iron availability for invading pathogens. We have hemoglobin. You're probably familiar with that one. It's located in the red blood cells, and it helps us acquire oxygen. There's also transferrin found in the blood and then in tissue fluids, and lactoferrin found in milk and saliva. And then there's ferritin that's found in all cells. But naturally, bacteria have evolved their own set of proteins that are designed to scavenge iron from our proteins. So it's a constant back and forth. But either way, iron is the rate-limiting growth factor for pathogenic invasions. So this brings us to the end of our chapter. To review what we've covered in this section, we were looking at the second-line defenses. They involved inflammation, phagocytosis, the interferons, and then the complement system.